Hello, I am Tetianna Smith Drysdale. We're going to be doing a presentation on my research, but first, a little bit of background knowledge on me. I am a junior working in the Hallett Lab. I am an environmental science major and a science communication minor. I have a broad interest in ecology and soil science, and I hope to further my interest in graduate school. So my research project is titled The Mycorrhizal Response of Bromus hordeaceus to Water Stress and Neighbor Density. So I started off my research project working with a graduate student within the Hallett Lab, and she was interested in exploring the facilitative relationship between the invasive grass species Bromus hordeaceus and a legume plant species called Acmispon americanus. For me, as I worked with her, I developed new research interest in Arbuscular Mycorrhizal Fungi, and this is a very important fungi in ecosystems across the globe, as it is able to form symbiotic relationships with about 80% of all vascular plant species. What it does is that it uses its small hyphae, or otherwise known as tendrils, to uproot nitrogen and phosphorus and other nutrients in exchange for carbon from the plants. And so because its hyphae is smaller than the roots, it has an easier time uprooting both nutrients and water. Now, it is well known for its skilled ability to help plants alleviate plant stress from drought, pathogens, or salinity. And so my questions were, I actually had two of them, and the first one being, do legumes change the rate of mycorrhizal colonization in neighboring plant species? With a hypothesis of the presence of a neighboring legume will reduce AMF colonization through an increase of nitrogen availability. This is because that's what legumes do. It is assumed that the Acmispon americanus would fix nitrogen that would increase the nitrogen in the soil environment. And my second question is, how does drought change mycorrhizal colonization rates, and does this affect change with legume presence? My hypothesis being AMF colonization rates will increase under low water levels. My methods section, so for my methods section, we will talk about the greenhouse experiment that I helped with, and then I went on to quantify AMF within the lab. So I started off working with a grad student, as I said before, in the greenhouse. Once the plants were grown to full adulthood, as you can see in this image right here, we took these different plants and we put them under a very powerful wash that removed just about all soil from them. And then we stored them under a 50% ethanol solution for preservation. Over the course of the summer, I proceeded to take the plants through a very tedious staining and destaining process. I took each plant from each treatment and I cut them up into 10 pieces. Then I proceeded to take those 10 pieces and I would put them through a staining process that involved a black ink called Scheffer's black ink. I proceeded to take them through a destaining process that included boiling them in potassium hydroxide and using a vinegar solution. Once those 10 roots were destained, I would take them, put them on a kind of like microscopic glass lining, and I would take them through, or I would analyze them underneath a microscope. And so here we have a left and right image of what I was and was not looking for. On the left side, you will see that there are these very dark lines, and this was the purpose of staining because the roots would otherwise be very difficult to see. So the ink would attach itself to the nodules and the hyphae of the AMF, making it easier for me to mark fungal colonization or no fungal colonization. So all these little lines that you see here, that is the hyphae. And any sort of nodules or any sort of circular shape, that can be assumed to be the actual nodules of the AMF. On the right side, where the arrow is pointing, there is no colonization. It is just straight root with very little activity going on. In the corner of this photo, you will see a little bit of colonization, but it's not as much as what was going on the left. For my results, they were actually contrary to my hypothesis. There was no major change in AMF colonization across all treatments, so that means that the different densities and the different water levels did not change AMF colonization. So on the left side, you will see an image that shows the 3 different types of Acmispon densities that we were measuring. 0 meaning no Acmispon americanus, 12 meaning a medium amount of density, and 24 being high amount of density. You will see here that the percent colonization is the y-axis and the density is the x-axis. And there really isn't too major of a difference across densities or percent colonization. So the same thing applies for the right image, with water level being on the x-axis and percent colonization being on the y-axis. There is no major difference between high and low. And so there are a couple reasons why this may be true. The first thing being that we were only doing this experiment for 1 year. And because of that, the legumes may have not had the ability to contribute as much nitrogen in the soil environment as it would if it had multiple years. This is because legumes fix nitrogen across their lifespan, but during their first year, when they do it, they're doing it for themselves, and they don't experience leaves falling off and decomposing to add that nitrogen-rich biomaterial to the soil environment. The second theory may be that Bromus hordeaceus just may not have that much of a dependency on the fungi. This is called a low mycorrhizal dependency. Bromus is a C3 grass. C3 grass just being the way that it photosynthesizes and fixes its glucose. And a lot of C3 grasses have a lower mycorrhizal dependency than, let's just say, C4 grasses. Regardless, this information is important because of AMF may help mitigate plant stress during the more frequent and severe droughts that occur in California grasslands. That is where these plants are from. And with climate change on the horizon, it is very important to understand how plants operate in these conditions. I would like to thank the people who I worked with on my experiment, Dr. Carmen Watkins and Dr. Lauren Hallett. I'd also like to acknowledge the Ronald E. McNair program that helped fund my summer research and beyond. I would also like to thank some people that supported me, such as graduate student in the Hallett Lab, Ethan Torres, Aiden Foster Green, Julia Albertson, Charlie Ellis, and Heather Dawson. So thank you so much for all of this support.